Hi, Mark. How you going?
You got Makeler in here now?
Say that again?
You got Makeler in here now? Okay, cool. Might be some of these stay here.
Yeah, I think that's right. Yeah.
How are you travelling at Parramatta?
Yeah, I mean, we've settled our research lab.
You got space down there? Or?
Yeah, we've got plenty of space.
Right.
It's good. Actually, more space than what we had at Campbelltown.
Okay. And I heard who's coming to talk to you?
Yeah, I don't know. Like, I actually don't know who's so. We definitely don't have issues, so I actually don't know who yet. I don't know. Yeah, so we've kind of been singled out.
Which is the trouble with those surveys because they say you're not, but then they do.
Yeah, and then it's
not
like
appropriate.
this whole list of people, and you're
Yeah.
thinking, how is this anonymous?
Yeah, exactly. Exactly right. Are you going to last the semester or are you going to...
Well, I have only till the end of the month, so I won't do I'll do this. Yeah, a
Right.
couple of
Who's
lectures.
taking over Makeler for you?
Oh well, Mark Temple usually does the lectures
Right.
for half of the unit, so
Right.
that's not a huge.
Right.
So it's not too disruptive actually.
Okay, I think we're going to get started. Well, there's anything on? No. Okay. Appear to be very loud. Okay, so this is the second lecture of molecular biology, and today we're going to have a bit of a look at what a gene is, locating a gene within a genome, and how we can use those data. So we're covering bits of chapter 7 and 8, and if you see a figure that's from a textbook, that is the chapter area in the textbook that we're covering. Okay, so this is the slide of the learning outcomes that I always put up, and essentially these dot points are what we're going to cover in the lecture, and you can use this again, as I said last week, as a revision. If you go through the dot points, you'll see the main or the salient features of the lecture, and you should be able to describe these things even for your final exam. Okay, so how we're going to start this is we're looking firstly at genomes on a large scale. So you can see if we look at chromosomes, the chromosomes of humans are on the left, and the set of chromosomes of mice are on the right. You can see that clearly they're both eukaryotic, so they have linear, both have linear chromosomes, and if we examine the setup of both of these chromosomes, both these sets of chromosomes, you can see humans, we've got 46 chromosomes, we've got 2 copies of 22 of each of the 22 chromosomes, and then we have our sex chromosomes. While mice, even though they're also higher eukaryotes, quite complex, they have a different number of chromosomes. They have 40, so they have 2 copies of the 19 chromosomes, and then they have their sex chromosomes. So even at the point of very higher eukaryotes, like comparing humans and mice, you can see that even how the genome on a large scale, so the genes on these chromosomes are slightly differently arranged. If you then take specific chromosomes, so we're now looking at, for example, the example here is human chromosome 9, and each chromosome has got this set of genes allocated to that chromosome. So human chromosome 9, it's got these set of genes. Doesn't really matter what they are for the purposes of explaining this. If we take another mouse chromosome, so not mouse chromosome 9, mouse chromosome 2, you can see that it's got the same genes located on that chromosome 2 that there are in humans. So you can see that there is genomes of closely related organisms. Even though the chromosome number is different, the genes are arranged in the same order along the chromosome. And this is, you'll see, if you see this in the textbook or when you read papers, this is described as synteny. So these genes being arranged in the same order, but it can be different chromosomes from genomes of closely related organisms, but they keep this same gene order. Okay, so and we're going to have a bit more of a look at the human genome as an example of the whole genome of an organism and what it's made up of. So if we have a look at, so again, this information is from the fact that we now have a genome sequence of the human genome, so we can actually do these kind of studies. So before if you don't have a full genome sequence of any organism that you're looking at, you can't do these kind of comparative studies because you don't know what's in there, what's in the genome of an organism, unless you can sequence all of the DNA. Okay, so this is a pie graph of the human genome. And essentially, let's have a bit of a look at what the genome is made up of. You can see nearly half of the human genome is in this grey bit. And this grey bit essentially are made up of transposable bits of DNA. So bits of DNA that move around, called transposable elements. They're kind of a molecular parasite. They insert themselves into the genome of essentially every organism has these transposable elements. And many of the genes that are in these transposons contain genes that encode proteins that catalyze this transposon process, so this moving around process. So you can see they represent nearly half of the human genome are made up of these bits of DNA that can move around, which is very interesting. If we then have a look at the coloured bits, so we can see straight away there's about a quarter of our genome are made up of the intervening bits of DNA. So if you have a gene sequence, we'll talk about this in more detail in this lecture, and then Mark will pick it up later on. A gene sequence in the genome will be made up of regions of the gene sequence that gets made into a protein. They're called exons, and then there are regions in between these exons that don't get translated, don't get made into protein, and they're called introns, and they're intervening sequences, and these bits need to be removed from the gene sequence before we can make that protein. You can see that about a quarter of our genome are made up of these these intervening sequences between exons in a gene. We can see also that there are other type of sequences that are repeated, duplicated, or repetitive. So things like sequences in the centre of a chromosome, so the centromeric sequence, or the ends of chromosome, which are telomeric sequence. They're repetitive bits of DNA. They make up a large proportion of our genome as well. And what you can see, hope quite clearly, is this very small piece of the pie, which is about 1.5% or so of all of our genes are protein coding genes. So this tiny proportion of the human genome is where all our genes that encode all the proteins that do anything in the cell. So it's a very small proportion of the rest of our genome are actually the ones that do everything or do a lot, or all of the processes that, even if you were just in the metabolism lecture, Mark talked about all the different metabolic processes, and these are all the proteins that do the stuff. And so they only make up a very tiny proportion of all of our genome. So if we then take this, where's my pointer, take this small part of the pie graph and have a look in more detail at what genes are in these protein coding genes, how they're made up. Again, we can only do this because we have the genome sequence of a lot of annotated genes that we can say are specific types of that fall in specific categories or families of types of genes that make those specific proteins. Okay, so first of all, let's deal with the fact that there's nearly 40% of our protein coding genes, we don't know what they are. Okay, so there's still a lot to discover as to what these protein coding genes are in our own genome. So for example, about 5 or 6 years ago, we discovered a new gene, therefore a new protein that gets made from that gene that is involved in DNA repair. So before that, obviously that protein still did something in the cell, we just didn't know about it. Okay, so we discovered this new gene that makes this new DNA repair protein, and it's quite crucial for humans to repair their DNA to have this gene and therefore this protein, but we didn't know about it before. Okay, so it was there, but we didn't know about it. So there's still a lot to discover about the genes, the protein coding genes in the human genome that we actually don't know anything about yet. So lots to still discover. Okay, so you can see there's also a large proportion of protein coding genes that are enzymes. And this is not a surprise because enzymes make up the bulk of our proteins, of the proteins in the cell, that do all of these different catalytic processes that we talk about or in DNA processing, in metabolism, in lots of different, everywhere essentially. There are also a lot of proteins that bind nucleic acid, okay, so that bind DNA and RNA. That also makes sense because a large proportion of what the cell does is that it is making, replicating DNA, it's transcribing DNA into RNA, and it's making protein from that RNA. So you need a lot of proteins that can bind DNA and RNA to help perform and regulate these processes. So you can see, so we've got nucleic acid binding proteins, so it could be bind DNA or RNA, ones that regulate these processes. Transcription factors, these are proteins that bind DNA and regulate the expression of other genes. So you can see they make up a large proportion of the protein coding genes. And you can see there's also other ones, obviously immune proteins, integral membrane proteins, etc. Okay, so this is the essential kind of setup of the types of protein coding genes that make up this very small proportion of all of the DNA in our genome. Okay, so we can move on now and talk a little bit more about what is a gene, how we identify a gene as a gene, etc. So if we're going to use protein coding genes as an example, so remember protein coding genes are the genes that get made, transcribed into messenger RNA, and then get made into that protein that's going to do whatever it is going to do in the cell. Okay, so genes must include, if they're a protein coding gene, they must include the code for an amino acid. Okay, so in the DNA, there's going to be what we call the coding sequence. So the pretty much the gene sequence that we read, 5' to 3', where you've got this start codon. It's going to be then read by the translation machinery once we make it into RNA, into an amino acid sequence. Every triplet codon gets read into a specific amino acid. We make a new polypeptide chain. We make the protein from that original gene sequence. So genes must have sequences that control transcription. We'll talk about this in more detail, what types of sequences they are. So they're not part of the gene sequence that gets made into protein, but they're external to the gene sequence, and we'll have a bit of a look at that. Transcription, so transcribing DNA into the messenger RNA to then finally make the protein later on, is a very tightly regulated process. And Mark is going to spend at least one lecture talking about, after he talks about transcription and the process, talking about regulation of transcription, because it's a very big thing that occurs in the cell. So it's a very tightly regulated process, and it's always in response to some extracellular signal. Okay, so you know the idea of if you've eaten a donut, you need to break down that sugar that you've just eaten that donut, and therefore that extracellular signal goes and says, okay, we need all the enzymes that break down glucose. We need to turn all those genes on and make those proteins so we can then do that breakdown process. And so these are the signals that the cell uses to then tell, essentially to tell the DNA, hey, look, these genes need to be turned on. So this is a very tightly regulated process. Okay, so coding sequences of a gene, and we'll talk about this in the next slides. We'll have a look at some examples that I think I alluded to this already. So we've got the gene sequence. The bits of the gene sequence that code for protein are found in exons, so in expressed bits of the gene, essentially. And the ones that we want to get rid of between exons are called these intervening bits of DNA within the gene sequence, and they're called introns. The sequences that I alluded to just before about gene sequences must have a DNA sequence, not part of the gene sequence, that control its transcription, control it being turned on or off, whether you're going to make a protein from that gene or not. They're called regulatory DNA sequences, and we'll talk about these in more detail because they're very important to regulate whether you're going to turn a gene on or off. So turning a gene on means you're going to make the protein from that gene, and therefore some extracellular signal said you need that protein in the end, okay? And turning a gene off means that process, break down glucose, whatever it is you're doing, has finished, therefore you don't need that protein anymore. That gene needs to be turned off because you never make more protein than what you require for your process in the cell, okay? So this is this feedback loop that always occurs and this regulation that occurs in the cell. Okay, so regulatory DNA sequences, we'll look at an example of the next slide has an image so you can have a bit of a look. So we're talking about a gene sequence, okay? So it has a start codon and a stop codon, and this is your gene that gets made into a protein. Regulatory DNA sequences are essentially out from that region, so either before that gene sequence, so 5' or upstream of that gene sequence, or after it, downstream 3'. This is all the same thing. Okay, so it's part of the gene, the DNA sequence, where that gene is on, say, if it's a human, on the chromosome. So it's part of the total genome sequence. Okay, so regulatory DNA sequences are important because they help, well, the actual DNA sequence doesn't help regulate whether you turn that gene on or off, but the proteins that bind that regulatory DNA sequence will help turn a gene on or off. And those proteins are proteins that regulate transcription, and they're called transcription factors. So these are the ones that bind the DNA. They have a specific target, and they'll target that gene and help turn that gene on or off. So we've got hundreds of transcription factors. You saw from this pie graph that we looked at before, there's nearly 10% of our protein coding genes are actually transcription factors. So these proteins that bind these regulatory DNA sequences in DNA, we'll see that they're quite conserved, and that's what they do. So they have a target gene and they help turn that particular gene on or off. Okay, so a regulatory DNA sequence, then you will see it's a very short consensus sequence, which is a sequence that is conserved, and we'll have a look at a couple of examples of that. And they can act on a gene that's on the same chromosome. So I said, remember, we've got a specific order of genes on each chromosome, and the regulatory DNA sequence can be before or after that gene, and they're called cis-acting regulatory DNA sequences, but you can also have something that's quite further away that can still be able to regulate the expression of a specific gene, so even on a different chromosome. And that's because transcription factor proteins are very small, they can diffuse around the cell, and they can act on genes that are located on different chromosomes. And these are termed transacting transcription factors. Okay, so let's have a bit of a look at what this looks like. Okay, so here, let's put into perspective when you look at these types of things, because it's always the same. If you see this kind of sideways L-shaped arrow, this is always the transcription start site of your gene, so where the ATG start codon is of your gene. So where pretty much you start making your gene from, or transcribing, in this case, your gene from, and then also the ATG start site is where you start translating your RNA, making a protein from that. But here we're talking about the transcription process, so the first step. So anytime you see this L-shaped sideways arrow, it's always where the ATG start codon is, so the transcription start site of your gene. So you can see here the gene is here annotated in yellow. So this is our gene sequence. And you can see that there are regulatory DNA sequences that are labelled. You can see that they're after the gene, or they can be after the gene, so downstream of the gene or 3' of the gene. This all means the same thing. This is where these transcription factors and activators, so other DNA binding proteins, also called transcription factors or transcriptional activators or co-activators, they bind the DNA at specific DNA sequences that are contained in these regulatory DNA sequences. So part of the DNA strand, okay? You can see that they also can be before our gene of interest that we want to turn on, transcribe. So you can see that they're 5' to the gene sequence, before, upstream. These three things mean the same thing. Okay, so these proteins, so here they're designated blobs. They're not blobs, they're proteins. By binding the DNA at these regulatory DNA sequences, they have an effect on the transcription machine that transcribes that gene. So the one that actually transcribes that gene into messenger RNA that then finally makes the protein later on. Okay, so this purple bit just before the gene sequence we're going to talk about in a lot more detail in the next few slides. It's essentially the area where the RNA polymerase binds the DNA, opens up the DNA duplex, and starts the transcription process. And what you'll see, and you get already an idea here, is that it's not just the RNA polymerase that's involved in transcription, it's a whole load of other proteins. It's a little machine that transcribes DNA into RNA. Okay, so we'll refer to this region here just upstream of our gene sequence, or just 5', just before the start site, the transcription start site of our gene. This area on the DNA is called the promoter, and we'll talk about this in more detail, and especially this region in the DNA, this conserved sequence of DNA, it's called the Tartar box, and we'll come back and have a look at that. So all of the proteins that are involved in transcription are also called transcription factors. So they're specific transcription factors that always come together in this transcription machinery that then transcribes DNA. And you have these other transcription factors, so that are specific for specific genes that come in at these regulatory DNA sequences. Okay, and that's essentially what I've said. Okay, so let's have a bit of a look. So we're looking at this purple region just upstream of our gene that we want to transcribe. Okay, so this promoter region. So it's essentially a combination of a short DNA sequence that's located very close to the transcription start site of the gene. You can see it's just upstream of the transcription start site where the ATG start codon of our gene is. And this is where all the assembly of the RNA polymerase and associated transcription factors bind there and transcribe that particular gene into RNA. Okay, so you can see, in bacteria, we have a slightly more, less complex set. We still have a promoter region upstream. So here is our gene that we're talking about that we want to transcribe. So the coding region of our gene that we want to transcribe. You can see there's also a promoter region in bacteria, and bacteria is slightly different in the case that they have DNA binding proteins that can either activate the starting expression of a gene or stopping the expression of a gene. So it's slightly less complex than eukaryotic cells. So Mark will talk about how bacterial regulation occurs, and he'll look at different operons from bacteria. So bacteria can also have one promoter, one promoter turning on the expression of several genes. And this is an example that Mark will use, is the lac operon. So one promoter can turn on expression of, say, 3 or 4 genes that all need to be made at the same time because they need it at the same time. We don't have operons in eukaryotic cells. We have one promoter for one gene. Okay, so each gene has its own promoter, and it needs to be turned on by the transcription apparatus and a specific transcription factor. Okay, so we've got this promoter region. Here's our gene of interest, the coding region of our gene of interest we want to make into a protein. And you can see upstream of the promoter and the gene, we've got these regulatory DNA sequences in the DNA that transcription factors bind to regulate turning on and or off that gene, or downstream of your gene, you also have these sites. Okay, so so we've been talking about the fact that once we transcribe a gene, we transcribe the whole thing. So we transcribe a gene that has the coding region of the gene that gets made into a protein, but it had also these intervening sequences between the coding region that we need to get rid of that before we can actually transcribe that gene, that RNA, into a protein. So these are called, so here's our transcription start site of our gene, and it would get transcribed in this way. Okay, so we've got a exon, exon, exon. So these are bits of the gene sequence that need to be joined together to form the coding region of our mature messenger RNA transcript that then gets made into a protein. And then in between these exons, these regions, are intervening bits of DNA that need to be chopped out, essentially, and these exons joined up together. That's essentially the process of splicing, and Mark is going to also talk about that once he's talked about transcription, because splicing needs of the RNA needs to occur before you can actually translate that messenger RNA into a functional protein. Okay, so how do we create this? So when we're talking about removing introns, we're talking about making a mature messenger RNA transcript. So once the DNA is transcribed into messenger RNA, it's not mature because it still has introns that need to be chopped out. Okay, so that's what the splicing event does. And then once splicing occurs, these exons are all joined together. You then have a coding region of DNA that only codes for amino acids, and then that can finally be translated into a protein. Okay, so creating a mature messenger RNA transcript can also change the number of exons that you can have in a mature transcript. And this is the process that is called alternative splicing. So, for example, if we look at the example of exon 2, there is a region in exon 2. So the whole point of this idea is that usually we remove the intron. So this orange bit of DNA between exon 1 and exon 2, we need to get rid of it in the process of splicing, and Mark is going to talk about that. So we essentially chop that little piece of DNA up and join these two exons together to make the first bit of our mature mRNA transcript that will then get made into a protein. The same thing with the next intron, you would need to chop this intron out and then join up those two exons. But what you can see is sometimes you can get regions of the exon that end up being a bit of an intron instead. So it's essentially this grey bit of this exon, for example. That what would mean is that once you join up exon 2 and exon 3 and it gets cut here, you're making a slightly different protein from that same gene sequence, etc. So the idea here is that you can get different protein products from the same mRNA transcript before you actually splice it. And this is called alternative splicing, and you can generate diversity within a genome because a single gene can produce different messenger RNA products that they can then get translated into different proteins. They're very often related proteins, but they can be different proteins. And anyway, okay, so what happens? So we remove these intervening bits of DNA from our messenger RNA transcript, so something that we've just transcribed from DNA, the gene that we've just transcribed from DNA into RNA. We need to remove these bits that don't code for protein, essentially. So bits of DNA that don't code for protein need to be removed. Once we've joined up all the exons, then now we have a coding region of DNA, and each triplet codon, starting from our ATG start codon, will code for a specific amino acid in order, and how that gets made, then it gets made into a protein. Okay, so we refer to, once we have spliced all exons together and we create a reading frame of the gene, it's essentially this uninterrupted set of triplet codons, and each code for an amino acid. But you can actually get 3 possible reading frames because you have a triplet codon. So what happens is, the first, where did you go? Oh yeah. Okay, so you can see, the first reading frame, so we've got, once we've made that DNA into a mature mRNA transcript, we now have what used to be ATG in DNA, now is AUG in RNA. This is our start codon. Okay, so this always, once we translate that bit of DNA from that gene, we make a methionine. Okay, so all proteins start off with a methionine at the beginning. And then you've got the next triplet codon codes for a different amino acid, valine. The next triplet codon codes for a different amino acid, et cetera, et cetera. But if you hadn't annotated that gene, and you didn't, for example, see or know where the start codon was of that gene, you can then start reading further down. So you can start reading at the second DNA base in that triplet codon. So then you get a different reading frame. So you can see, rather than reading ATG or AUG as your start codon, you can read the next 3, UGG. UGG codes for a different amino acid. And the next set of triplet codons will then code for a different amino acid as well, et cetera, as you go down the line. And then you can also get a third reading frame from the same bit of DNA sequence, where you would start reading from this G rather than this AU. So that would mean you'd get GGU. GGU codes for a different amino acid, et cetera, as you go down the line. So it then becomes very important to know where your gene starts, so where your ATG start codon is, and where your gene ends, where your stop codon is. And this is important when we annotate genomes, because we really do need to know where a gene starts and stops to know what the reading frame will be of our final gene. Okay, so once you've found an ATG start codon of a gene, the next stop codon you find, the next stop codon you find would be where that gene stops being made, and therefore also where that protein stops being made once it gets translated. Okay, so genome annotation, one of the most important things for genome annotation is to identify an open reading frame. So essentially where the start and stop of a gene sequence is. Okay, so annotating a genome, so once you've got lots of DNA sequence from a particular genome of whatever species you're looking for start and stop codon, so with the location of different genes in that genome. You're also looking for other important DNA sequences that control expression of those genes. So these types of regulatory DNA sequences, often called elements, but part of the DNA, regulatory DNA sequences. These are also regions of the DNA that you can find because they're very conserved, and we'll have a look at some examples. Things also like origins of replication, so essentially where an organism starts replication. This DNA sequence is also a conserved region in the DNA, and you can find it using now with having so many genome sequences. We have we can very easily find conserved sequences of DNA that represent these types of DNA sequences. And things like telomeres, so the ends of eukaryotic chromosomes, those telomeric, those ends on the of the DNA are very conserved. They have a specific DNA sequence, and you can search for it and you can find that. Okay, so what's important is these types of functionally important sequences that I've talked about are very conserved between organisms, and they can be separated by millions of years of evolution, but they're still highly conserved regions of the DNA. And we can, after we have a genome sequence of an organism, we can identify these very highly conserved DNA sequences using specific programs. But before you can do that, you need to be able to sequence the DNA. So now what we're going to talk about a little bit is how you go about sequencing DNA. Okay, so this is a method that we'll go through now. It uses a DNA polymerase. So a DNA polymerase is used in cells all the time when a cell replicates itself. Okay, so that enzyme, again, this idea of using a tool in a molecular biology lab in vitro, we say we use a DNA polymerase. That DNA polymerase is something that occurs normally in a cell, and it's an enzyme that does perform essentially the function that we're using it to perform in vitro. It essentially replicates DNA. Okay, so once you need to replicate a piece of DNA, you need a DNA polymerase. Okay, remember, if you need to transcribe DNA into RNA, you need an RNA polymerase, etc. So all of these enzymes that we now use in the lab as a toolkit for molecular biology, it's only because we understand how they work in the cell in its normal environment. So DNA polymerase is always used in any cell that needs to replicate itself, every cell, essentially. And so this method of sequencing uses a DNA polymerase, and it uses chemically modified nucleotides. So in a normal polymerase chain reaction, which we'll talk about in more detail, you have to not only supply a DNA polymerase that will copy your DNA, make millions of copies of your DNA, of your little piece of DNA that you want to make in your polymerase chain reaction, you also have to feed it the DNA bases, the A, T's, G's and C's, or else that DNA polymerase can't make a new DNA strand if you can't give it the ingredients it requires to make that new DNA strand. Okay, so the nucleotides, we have to supply the DNA polymerase so it can make this new DNA strand in the case of DNA sequencing, also in the case of a polymerase chain reaction. Okay, so in DNA sequencing we use modified nucleotides, and we'll see how different they are and how normally what you use and how different the modified ones that we use for DNA sequencing. So this was the method that we're going to talk about, this Sanger DNA sequencing, was essentially the method to sequence the first iteration of the human genome that I talked about last week, so in 2001. And it took a long time, again, I'll say this again, lots of people and lots of money to sequence that human genome for the first time, okay? So using this Sanger sequencing method, it's a very work-intensive method, and one of the reasons is because you don't get very long reads of DNA, so you have to do a lot more work to get a DNA sequence of a genome using this Sanger DNA sequencing method. So now, or more recently, we use a modification of this Sanger sequencing method, and I touched on this last week. It's called next generation sequencing or massively parallel DNA sequencing, and this is now much easier to produce a sequence of a large genome, like a human, so you can go and sequence yourself, for example, and it's very rapid, very cheap because it's all automated. You don't need lots of people doing stuff, and it's essentially an improvement of the way that we sequenced before. So Sanger DNA sequencing we still use So when we have to sequence a gene that we've made in the lab, in our research lab, we send it for DNA sequencing, and it gets sequenced by Sanger sequencing. So this next generation sequencing is really only used when you've got lots and lots, big pieces and lots of DNA to sequence. You would use this next generation sequencing. So we're going to have a look at the original Sanger sequencing, and if you happen to do genes, genomes and human health in third year, Graham talks about the next generation sequencing in more detail, but we won't be covering it. Okay, so let's have a bit of a look at how this method works. So it was developed by, the reason why it has the name Sanger sequencing, it was developed by Frederick Sanger, so the person who developed it. And it's also known as the di-deoxy chain termination method, and I will remind you why that is in the next slide, because we'll see a few images of these modified DNA bases that are used in Sanger sequencing. So essentially the idea is that we incorporate these chain terminating modified deoxyribonucleotides during DNA synthesis, and in that way we can get a DNA sequence. So we're going to see how it works. Okay, so let's have a bit of a look at the normal, the normal deoxynucleotide. So the normal deoxynucleotide, got an example of thymine here. So remember, it was always have a 5' phosphate, sugar, deoxyribose sugar, and then the particular DNA base. Okay, so this is what's always given as the ingredient for the DNA polymerase. So A's, T's, adenine, thymine, guanine, cytosine, as a deoxynucleotide needs to be fed into a DNA synthesis reaction. Okay, so DNA polymerase to synthesize, to read the DNA strand that you want to sequence, in this case, it needs those DNA bases to then make the new DNA strand. Okay, so these are supplied in the reaction. Okay, so remember the DNA, it's deoxyribose sugar because it doesn't have an OH group that the normal ribose sugar has in RNA. Okay, so it's removed one oxygen, so it doesn't have a hydroxyl group at this 2' position in the sugar anymore. Okay, so the other important bit, which we'll talk about in the next slide, is that at the 3' position in the deoxyribose sugar, we do have a 3' hydroxyl group. This is really important for DNA synthesis reaction. So we need to have a 3' hydroxyl if we want to make a new DNA chain. And we'll see in the next slide why that is. So these di-deoxyribose sugar, so in these DDNTP, so di-deoxynucleotide triphosphate. Remember, this is a nucleotide triphosphate, so nucleotide, sugar, phosphate. These are these modified DNTPs that we use in the Sanger sequencing method. And you can see it's 2 times deoxy, okay? So di-deoxy, so it's removed twice the oxygen at these 2 positions. So neither of them have a hydroxyl group at these positions in the sugar. And that's important, and we'll see what normally occurs, and then how we incorporate these DDNTPs, so these di-deoxynucleotide triphosphates. Okay, so let's have a look at a normal DNA synthesis reaction. So we've got our DNA strand that we want to determine the gene sequence of. Okay, so our template strand, we often call that the template strand because a DNA polymerase also always requires a template, just like the RNA polymerase requires a DNA to read to make the RNA in transcription. A DNA polymerase, when it replicates DNA, also needs the DNA strand to read to make the complementary new strand. So in this case, for DNA sequencing, this is a strand we want to find out what the gene sequence is. Okay, so we don't know what that gene sequence of that strand is, but the DNA polymerase, with the aid of a primer, a short bit of DNA, that is complementary to the DNA that we want to find the sequence of. Once it can essentially hop on the DNA here and make a new DNA strand from there. So we can read this one, make a new strand above it, which is complementary. Okay, so you can see that so in a normal DNA synthesis reaction, we need a small primer that binds the DNA, that's complementary to the start of the DNA, and then the DNA polymerase obviously is added into the reaction, and you can see this hydroxyl is this 5' hydroxyl that we talked about here in the normal DNTP. Okay, so we've got an incoming DNTP, so the next one along, so it's a C in the DNA strand, the template DNA strand. The new DNA strand, we need to put the complement, so the G. Okay, so this incoming DNTP gets joined up to the new strand that's being made via a nucleophilic attack on this phosphate by this hydroxyl group. Okay, so by this 3' hydroxyl group essentially in the DNA base before it. Okay, so we've got this incoming DNTP that was sitting in the mixture because we need a G there because we've got a C complementary to that, and so then we'll get a phosphodiester bond occurring here once this reaction occurs and these phosphates go away. And then that would happen the same thing. So this 5' hydroxyl will then form a phosphodiester bond with the incoming phosphate from the next DNTP. So if there's a T there, this one would be D, the next one would be a DATP, so it's an adenine. So thymine always base pairs with adenine, so then we would have an incoming adenine into this reaction. And then as you go along, you continue to make the new DNA chain by this same reaction. So we always need a 3' hydroxyl to be able to make a phosphodiester bond, so a new growing DNA chain. Okay, so that's what happens normally. So what happens during the Sanger method of sequencing, we have the normal DNTPs, so the normal ones that we've just talked about, where they have a 3' hydroxyl, we can make a new DNA chain, and they're in a certain concentration in the reaction. We also have the modified DNTPs, so these ones that don't have a 5' hydroxyl here, and they're added into the reaction at a very low concentration. Okay, and so what happens is that clearly once this gets incorporated here, so we've got a T in the chain that we're trying to find the DNA sequence of, clearly in the growing new chain, we need to put an A there. Okay, so if this modified DNTP gets incorporated here, okay, we can form a phosphodiester bond with this DATP or DDATP, but then the next one along we can't form any bond because we don't have a 5', a 2' hydroxyl, or 3' hydroxyl here anymore. We've got a hydrogen. Okay, so the next one along we can never add a G at this point. So once we've added the A, this pretty much terminates synthesis of the DNA at that point. And so by carefully controlling concentration of normal DNTPs, so these ones that we normally, that make a new DNA strand, and these modified DNTPs, so these dideoxynucleotide triphosphates, on average, every molecule of newly synthesized DNA will incorporate one of these dideoxynucleotides in the sequence and terminate DNA synthesis at that point. Okay, so just before we go and have a look at what happens and an example of how this sequencing occurs, to reiterate the fact of the fact that the Sanger DNA sequencing is a slightly modified version of the polymerase chain reaction of what you'll be doing in the lab. Instead of using 2 primers, so normally for a polymerase chain reaction, and you've all done your computer prac, so you saw to make this bit of the TP63 gene, you had a forward primer, so it was the start boundary of the bit of the gene you want to make, and you always have a reverse primer, so the end boundary of the bit of the gene you want to make. Okay, so that's a typical, you always need a forward and a reverse primer in a typical PCR reaction to make a gene, to amplify a gene. In the case of DNA sequencing, you still use the DNA polymerase, but you actually only have just 1 primer. You're not making the 2 strands of the DNA of your gene. You're essentially reading the strand that you don't know the DNA sequence of and making the complement, and that's the one that you then have the sequence of, and then you can see what sequence your original gene was. Okay, so we only need the sequence of 1 strand of the DNA to be able to know what the sequence of our unknown thing is. Okay, so let's have a bit of a look at how we would Sanger sequence this piece of DNA, and then we can have a little bit of a break. Okay, so we've got a bit of DNA, it's obviously double-stranded. We only sequence 1 strand of their DNA, so we'll have in this case we have a primer in a known region of the DNA of the gene that we so a known region of the gene. You can also have primers when you don't know what the gene sequence of what you're looking for, so it'll be slightly different. But in this case, we've got a small piece of DNA that is complementary to the start of the bit of DNA that we want to find the gene sequence of. And you can see it's complementary, T base pairs with A, as we go along. Okay, so at this point, this is the basis that the DNA polymerase can hop onto the DNA and start reading this strand and adding the complementary strand on the top. Okay, so these incoming DNTPs will then they're the ones that will be fed into the reaction and we can make our new DNA strand. Okay, yep, we already saw that we require this 3' hydroxyl from a normal DNTP to be able to do that. Okay, all right, so what happens then with the Sanger method? So here we've got our primer, so the piece of DNA that we've put into the reaction to start off the reaction in the first place. Then we've got our newly synthesized bit of DNA in green. So we can see that the DNA polymerase is in this region across here. C has put in a G has put in a C, etc., etc. And because we have a very controlled amount of these modified DNTPs, at some point in that whole DNA synthesis reaction of that particular strand, you're going to incorporate one of these dideoxynucleotide triphosphates at the end of obviously it's at the end, because once you incorporate these modified DNTPs, you stop DNA synthesis at that point. So this essentially creates a population of fragments where each base of new DNA fragments that you've synthesized that terminates with one of these base terminating DNTPs. And you can see over the course of the whole reaction, you will end up with, at every spot once you've made a new DNA strand, at every position of the DNA strand, you'll end up with one of these red DNTPs. Okay, so, and essentially once you have at every spot along here one of these, you can actually read them off, okay, in the machine that you use to do DNA sequencing. Because each of these dideoxynucleotide triphosphates, when you're fed into this reaction, they actually have a tag. They have a fluorescent tag. They have a specific, so an A DNTP or DATP has one fluorescent tag. The guanine version has a different fluorescent tag. The cytosine version has another one, and the thymine has another one. And so, and lasers can read the frequency of these different tags. And so it essentially, by using, so it's a very automated process, so you don't actually do it yourself. The machine does this, but in any case, at each point in that DNA sequence, you'll have one of these that has incorporated the DNA synthesis reaction has incorporated one of these, and you can actually read what the sequence is at each point in the DNA sequence, and you can detect that sequence readout in this type of format. So you can have so in the end, what you end up with is an output is here's your DNA sequence, and you can see at each point in your DNA sequence, you will have read one of these fluorescent dideoxynucleotide triphosphates that's been incorporated. You can see a green peak indicates that you've got an A there. Another green peak indicates you've got another A. A red peak indicates you've got a T. So this is the identity of the different fluorescent tags that are used for the different modified nucleotide triphosphates. And then in the end, you can get a whole sequence of that piece of this blue piece of DNA that you originally wanted to get the sequence of. And so the problem, or not the problem, but one of the limitations of this sequencing reaction is the length of the DNA that you can sequence by this chain termination method. And if you get a good DNA sequencing, what we call a DNA sequencing read, maybe it might be 1,000, 1,500 base pairs from one DNA sequencing reaction, and that's about it. You'd have to do many, and in fact, that's what they did for the human genome, many sequencing reactions to cover the whole of the human genome to be able to get Sanger sequencing for example, the whole human genome. So it was a really big feat to actually sequence the human genome using this Sanger sequencing method, and now this next generation sequencing makes it so much easier to be able to sequence large pieces of DNA. But when we need to sequence a gene, so in the lab, we always use this, well, we don't do it ourselves, you send it out to do it. It's all automated now, companies do it, you send your gene, your DNA to be sequenced, and you end up with essentially one of these reads being sent to you, and you can determine what this gene sequence of your thing that you're trying to determine what the sequence is. Okay, so if you want to have a look at an animation of the Sanger sequencing method, if you go to that website, it should work. We're not going to do that now. So I think we'll have a little bit of a break. Maybe we'll start at 10 past again, or whatever. About 10 past 1, and then we'll talk about what we do with the DNA after we've got all this sequencing information. Okay, I think it was about 10 minutes. Hopefully most people have come back. Okay, so now what we're going to do is, we know how we can get sequence of DNA from genomes, for example. And what we can do is once we have these data, they're very powerful for us to be able to study the conservation of DNA sequences between species. So we talked briefly before about these consensus sequences in DNA. So things like the promoter region of DNA and things like that. These, what you'll see, we'll look at some examples. They're very conserved across all different species, okay? And we can only do these types of studies if we have lots and lots of genome sequences from different organisms to be able to do these studies with. Okay, so I've started here with a slide about vertebrate phylogeny. So you would have seen a phylogenetic tree before. So this is essentially a phylogenetic tree to help us identify nearest relatives or common ancestors. So we're going to use some examples from this phylogenetic tree and have a bit of a look at the DNA bits of a DNA sequence from these organisms. Again, once we have DNA sequences from a whole load of different species, we can actually make a phylogenetic tree based on genetic information, okay? So it's not just a phylogenetic tree that someone has decided to put together, it's gene sequence, okay? So we've got gene sequence for all of these different species, and based on conservation between different gene sequences, we can actually make a tree to identify common ancestors or nearest relatives. Okay, so we're going to use a few of these different different species from different areas of the phylogenetic tree. So amphibians, birds, we're going to use dog, cat, etc. Okay, so here, what I've put up here is, I know it just looks like a list of A, T's, G's and C's. This is what's called multiple sequence alignment. So we're looking at a small region in the DNA, in the genome of one of these organisms. So you can see the first one, so the first line is from the human, okay? And this line continues to the next line of human, okay? So unfortunately, if we had a big long piece of paper, we could actually show that all in one line, and each one of those species that you see underneath would also be all under the same line, but we can't do that when we show it on a piece of paper or on a slide. We need to go to the next line, okay? So effectively, every time you see human, so we're pretty much the DNA sequence, so a bit of a read from the genome of the human goes across here, and then it goes to the next line where it says human, and then it goes to the next line where it says human, and it goes to the next line where it says human. Okay, so that's just this little bit of DNA that we're looking at from the human genome. And we're comparing it to the same area of the genome from the mouse. Okay, so again, the mouse is always the second line, and as you go down, it continues with the mouse, and etc. So we've got rabbit, dog, and etc. These xenopus, etc. Whoops. Okay, so this is what's called so we're comparing DNA sequences from multiple related organisms, and what we can start seeing once we do these types of comparisons is that we can start to identify sequences that are highly conserved between different organisms. That can be and if they're conserved, we can start saying that they're conserved through evolution. Gee, they must be an important bit of the DNA of the genome of that organism if they're conserved across evolution. Okay, so so you can see there's also regions of this gene sequence that are not coloured, and then regions of that sequence that are coloured. And hopefully what you can see is that the coloured bits of the DNA sequence that we're looking at is also very conserved across all of these species. Okay, so you get tracks that are nearly exactly the same. You can get some anomalies, but there's a big piece here in the coloured A's, T's, G's and C's that's very conserved across all these different species through evolution. So this is likely to be a gene sequence. That's probably what it is. It's quite long. Little consensus sequences of DNA, like these regulatory DNA sequences or a promoter sequence that we'll see an example of, they're much smaller regions of DNA that are conserved. But once you get a big block like that, it's likely to be a gene sequence. And you can see that gene must be pretty important because there's blocks of it that are nearly all conserved across all these different species that we're looking at in this multiple sequence alignment. Okay, so we can start and you can do this across, there are programs that do this, and you can do this across the whole genome of, say, one organism and all these other ones. Okay, so you can look at lots and lots of gene sequences or the whole genome and compare it to the whole genome of another species. So you can do a lot of this type of comparison in sequences. Only once you have the genome sequence of an organism can you do these types of things. Okay, so we can view sequence data like this. So this is the gene sequence of a bit of the region of the genome of all these organisms, but we can only really see a small bit of the gene sequence. Okay, so we can see, you know, one bit. Okay, so it's kind of limited in what we can look at. We can convert these kind of sequence alignments into a graphical representation where the height of the graph and the breadth of the peaks on this graph denote whether you've got a region, how long the region of similarity is. Okay, so we'll have a look at an example. So these are the types of graphs that I'm alluding to. So again, we've got on the y-axis, we've got the DNA sequence. Okay, so we can't see what the DNA sequence here is because it's much more compressed, but this is the DNA sequence of the region we'll be looking at. Okay, so you can see that there's DNA sequence, there's a promoter always, the gene is always going to have a promoter if it's from a eukaryote. This is our transcription start site of our gene, so where our ATG start codon is. There's some sequence. Here's an exon in our gene sequence. There's an intron. There's another exon, intron, exon, etc. And then we can continue this graphical format for as long as we like. Okay, so we've got these peaks, and essentially the width of the peak denotes the DNA sequence. So the length of the DNA sequence that is similar. So essentially, like a block of this stuff that is similar, okay? So the A's, T's, G's and C's that are similar, all in a line. And the height of this peak, which is the x-axis, is telling you how conserved that region of DNA is, okay? So if there's a lot of conservation, then obviously the peak will be big across that piece of DNA, okay? And hopefully what you can see quite easily is when you have conservation in gene sequence, especially in exons, so in bits of gene sequence that codes for protein, for making the protein, we get lots across that whole exon, we get lots of conservation. So essentially we would be seeing something like this if we're looking at the actual DNA sequence, okay? So we get big blocks of conserved or nearly conserved DNA sequence. And the same can be said for all the other exons. You can see that there's huge conservation. You can see that there's also areas that aren't conserved. Introns, they're not conserved. The DNA sequences of intronic DNA, of intervening sequences between exons is not conserved at all. And in fact, you can see that it's not conserved at all. There's no peak there, okay? And that corresponds exactly to where an intron is. You can see there's also other areas of the DNA that are conserved, okay, so that they have some conservation, and probably not as much as gene sequence, exons in a gene sequence, but that's quite highly conserved. And they would be regulatory regions of DNA that are important to facilitate expression or turn on or off expression of a particular gene. So a promoter region, there's quite high conservation, and we'll have a look at an example of a Tata box, and you can see that. And there's also other regions before, upstream of our gene, and downstream of our gene that look like there's some conservation there. And they would be other regulatory DNA sequences where transcription factors will bind and help regulate that gene, turning that gene on or off. So you can see just by having a look at these kind of simple graphs, you can see regions of the DNA that are conserved when you're comparing lots of different species together. Okay, so regulatory DNA sequences, like I said, like things like where the promoter binds and these other regions that are outside the gene sequence are very conserved through evolution. And these regions, and I want to just reiterate the idea of this because it's a very important factor here, that regulatory DNA sequences are essentially where transcription factors bind, and transcription factors bind to the DNA. So if you remember from last week, we had a slide where it showed the major groove of the DNA double helix, and this is a region, so it would be a specific DNA sequence in the major groove of that double helix where that gene sequence, close to where that gene sequence is, that this transcription factor binds, that then regulates whether you're going to turn that gene on or off. Binding of transcription factors to DNA is dependent upon very specific interactions that the transcription factor protein makes with the DNA. Okay, so once you start to, then it often involves hydrogen bonding and very often involves things like positively charged amino acids on the transcription factor protein binding the negatively charged DNA backbone on the DNA. And these interactions, these protein DNA interactions, are very highly conserved because transcription factors only bind a conserved consensus sequence in the DNA, so these regulatory DNA sequences. And so you get conservation at the protein level, so at the transcription factor level, so specific amino acids are very important for that transcription factor protein to bind that DNA, but also that DNA sequence, that regulatory DNA sequence is also very highly conserved, so that transcription factor can bind the DNA. You start changing either of these two things, transcription factor can't bind the DNA anymore, and then you start getting issues. And a great example of that is the TP63 gene that you're going to work on in the labs, where you have a TP63, the transcription factor, if you recall, it binds the DNA, but if you make that mutation that you did when you did the Euclin-Pruder-Prac, this mutation from a positively charged arginine residue to a polar glutamine, so this R204Q mutation in the protein, that transcription factor can't bind the DNA anymore where it's supposed to, and then you get issues. So obviously that transcription factor can't turn on expression of whatever gene it targets, and you get this developmental disorder. Okay, so then there are knock-on effects, whether you change the DNA, if the DNA gets mutated, or whether you change the transcription factor protein that binds that regulatory sequence in the DNA. If you start changing that, then you can also have issues. Okay, so the example that we're going to use is this P63 protein, which is the same protein that, essentially the same protein that we're working on in the pracs. So this P63 protein, it's like the tumour suppressor P53, that is our main tumour suppressor. In sea urchins have P63-like genes. Well, you can see humans, we have 3. So we've got TP63, which is the one you're working on in the lab. We've got the tumour protein 53, so our main tumour suppressor protein, P53, and then also P73. So we've got these 3 genes that essentially means that in evolutionary terms or in evolutionary time span, humans seem to have increased the gene number for that type of protein. So clearly there's some reason for humans to have done that, and the importance of these genes and the proteins that you make from these genes have a clear importance, and they're all going to be binding DNA and helping to express specific genes. Okay, so if we have a look at the protein sequence of these P53-like proteins, so before those lines that we were looking at were A, T's, G's and C's. So it was DNA sequence that we were looking at before in that multiple sequence alignment. So now what we're looking at, we can do the same type of multiple sequence alignment and look at the protein sequence. So look at the amino acid sequence of a particular protein that we want to compare to other similar proteins or other proteins, similar proteins from another species, which is what we're doing here. So we've got some human ones, the P63, like the one essentially the protein of the gene you're working on in the lab, and then we've got the sea urchin ones as a comparison. So you can see just we see an amino acid sequence or a bit of the amino acid sequence of this P63 protein, and you can see that there are regions again that are highly conserved between these similar types of proteins in humans. So remember, we've got 3 genes for these P53-like proteins, and we make 3 proteins from that. But they're similar, similarly conserved in different regions. In some places, they're exactly the same. There are also some conservation between the human versions and the sea urchin versions. So anything in red is conserved, okay? So across the line. The spots in black are essentially amino acids that are essential. These are all transcription factor proteins that will bind the DNA at a particular regulatory DNA sequence that will be conserved. All the spots are essentially amino acids that are important for binding to DNA, and you can see that's a lysine. So a positively charged amino acid binds DNA, binds negatively charged DNA. Makes sense? That's pretty conserved all the way across, all the way down. These P53-like proteins, you always get a lysine. Okay, that obviously is important for DNA binding. Serine, okay, serines will make a hydrogen bond. That's also a feature of DNA binding proteins that bind DNA. So again, you can see that there's a full conservation of a serine all the way down. Okay, so whichever P53-like protein you look at, there's always a serine in that position, in that protein, once a protein folds up and forms its structure. And again, you can see the other one. So we get another positively charged amino acid, an arginine. That's also fully conserved, okay, as you're looking through each different type of human P53-like protein, or even looking at the very far away sea urchin one. Clearly that needs those amino acids as well. So, so amino acids that are essential for sequence-specific binding to DNA, to the regulatory sequence in the DNA, are conserved, whether you're looking at the human version of the same gene or protein made from that gene, or the sea urchin version, separated by millions of years of evolution. Okay, so amino acids that are essential to bind DNA are conserved. Okay, so it doesn't matter what species you're looking at, what evolutionary distance, if they're the same type of protein, like these are, you will get the same conservation in amino acid sequence in that protein, because it's a transcription factor and it has to bind the DNA. And you can see the conservation whether you look at the gene sequence, so the DNA sequence, or that encodes this protein, or the protein sequence that is made from that gene sequence. So you can see conservation at both ends, and it makes sense when you study it. Okay, so we've been talking about conserved sequence in DNA. There are also, once you transcribe DNA into RNA, there are also conserved sequences in the RNA that are important for translation. So this is sequence in the bacterial RNA once it's transcribed. So before it's translated, there is essentially before the sequence that you've just transcribed of that gene in bacteria. There's what's called a Shine-Dalgarno sequence. So it's a region in the RNA that is required to assemble the ribosome on. So just like there's the promoter region that's required at the start before a gene sequence to assemble the transcription apparatus to transcribe that gene into RNA, same goes for when you've made RNA. The ribosome has to bind somewhere. It has to know that it has to start to translate that RNA into a protein somewhere as well. And that's usually just before the RNA sequence. We have the same sequence in eukaryotic messenger RNA, and it's called the Kozak sequence. And again, it's where the ribosome assembles to then find your AUG codon that the ribosome will put the first methionine in your protein and then continue to translate your protein. Okay, so these regions also in RNA are conserved as well. Okay, so let's go back now and have a look at this, this promoter region, this very conserved region in the DNA that is just upstream from a gene that you're going to transcribe. Okay, so this promoter region is essentially where this whole transcription apparatus binds and then starts the transcription process, and RNA polymerase is the main thing that reads the DNA and into RNA. Okay, so here we've got our sideways L arrow. So this is the transcription start site of our gene. Okay, this is where our ATG start codon is of our gene. You can see it's designated as +1 here. So anything before that will be a minus number, and anything after that will be a plus number, okay? So essentially the promoter region of that gene is going to be upstream or before the gene sequence, and it's usually somewhere around 30 base pairs before that this transcription start site of a gene sequence. And that's also quite conserved, how far away a promoter region is from the start of a gene sequence. It's not going to be very far away. It's around 30 or so bases before the start of the transcription start site of a gene. You can see that the thing in purple is this tartar box. So each promoter has this region in the DNA that's very conserved, and we'll have a look at how conserved that is. It's called a tartar box because it's rich in TA base pairs in that bit of DNA. Remember, a gene is always double-stranded. DNA is always double-stranded. So you're always going to have TA, in this region, lots of TA base pairs. In fact, it's called a tartar box. And this is the area where the RNA polymerase binds, opens up the DNA, and then starts transcription occurring. And again, the idea of a tartar box being rich in lots of A's and T's is a region of the DNA that you can open up much more easily. So remember last week we talked about if you have an AT base pair, you have 2 hydrogen bonds. If you have a GC base pair, you have 3 hydrogen bonds. It's much harder, or takes much more thermodynamic energy to break open a GC base pair. But if you have lots of AT base pairs all together, all one next to each other, you end up with a region of DNA that is much easier to break open. And in fact, that's where the RNA polymerase essentially breaks open the DNA. Remember, can't do anything with double-stranded DNA, needs to break open to be able to transcribe that DNA into RNA. Okay, so let's have a look at the DNA sequence at a Tata box at a promoter region. Okay, so if you align the DNA sequence in this Tata box region, so around this transcription start site, about 330 bases up from a transcription start site of a gene, and if you align, say, 100 different promoters, in this case, this is the data from 100 different promoters, you can identify a consensus DNA sequence, so a consensus, a recurring theme in the DNA sequence at a Tata box. Okay, and clearly we need the Tata box to start transcription occurring. So here we've got, the DNA sequence. We have a transcription start site of our gene. You can see it's designated +1. This is now not a sideways shaped arrow, but still an arrow. Okay, so this is where our gene would start. Okay, so the ATG start codon of our gene. And about 30 or so base pairs up, in this case it's 35 base pairs before the start site of our gene. Whoops. Where are we? You have this Tata box, so this consensus sequence. So how did we arrive at this consensus? So let's have a look at the consensus sequence first. So it's TA, PA, okay, you might have an A or a T there, but it's very rich in A's and T's, okay? So you're going to have lots of AT base pairs in that region, in that double-stranded region of that DNA. So how do you arrive to that? So you can see essentially what you have here is what is the frequency. So after looking at 100 different promoters to identify a consensus sequence from a Tata box, what is the probability that you will have an A, a C, a G, or a T at these positions? Okay, and so you can see probability to have a T, 83%. Okay, so most of the time at minus 35 bases, so 35 bases up from your start of your transcription start site, you're likely going to have a T. And the same would be the next one along. So 90% of the time you would have an A. 100% of the time you'd have a T. 95% of the time you'd have an A, etc., etc. Okay, so you can get this information by looking at hundreds of different promoters from also different species, and you can identify like this a consensus sequence for the Tata box in a promoter. Okay, and so this doesn't vary from this sequence. Doesn't matter what species you're looking at, and so this is quite highly conserved, and clearly it's obviously an important part of the whole transcription process is and you can identify the Tata box very easily upstream of your gene of interest when you annotate a genome. So if you find a gene 30 bases up, there'll be a Tata box. And then there'll be other regulatory DNA sequences that you can also identify based on sequence similarity and therefore sequence conservation between other species using lots of different programs, bioinformatic programs. But with all of this genome information that we have, it's very easy to find this information and to annotate a genome accordingly with all of this information. Okay, so that's what a consensus sequence looks like and is. So it's a region, a small region of DNA that is conserved across lots of species and it's important for function. Okay, so just before we move on to the next few slides, the last few slides I should say, so what parts of our genome would you expect to be most similar to the genome of other organisms? Any of the ones that we've been talking about? So yeah, so that would be when we're talking, yes, the protein coding region. So the exons in a gene sequence. Is there any other regions that might be similar that we've just talked about? The Tata box. So the promoter region of a gene is going to be very similar because it will have this Tata box and things like these regulatory DNA sequences where other transcription factors will bind. They're going to be also highly conserved across the genomes from other organisms. Okay, so the last few slides we're going to have a bit of a look at how we can use sequencing data from similar species to determine, for example, ancestral, so what the ancestral sequence of a species might have looked like. And so we're going to use humans and chimps as examples. So the human genome is very closely related to other mammalian genomes along large segments of chromosomes. Our closest biological relative from the genome sequence, okay, so we're comparing one genome of human, or many genomes of human, genome of chimpanzee, okay? And if you compare those genomes, that's our closest biological relative, and we share a common ancestor about 7 million years ago. So genetic differences between humans and chimpanzees can range from large-scale rearrangements in chromosomes to single base changes, and at the end of our lecture we'll be talking about these single base changes that are called single nucleotide polymorphisms, but we'll come back to those. Okay, so the top here was showing you humans and chimpanzees can differ at a single base pair. Okay, so they're very small differences between those two organisms, or very large differences, things like errors in replication, or large differences that could be the cause of it could be caused by recombination or duplication, so rearranging parts of chromosomes. And a nice example of that would be, for example, if you looked at human chromosome 2, you've got this line of genes, okay, in chromosome 2. You can't see what they are, but you can see there's a specific order of genes on human chromosome 2. And in the chimpanzee, this chromosome 2 is now in two pieces, okay? And you can see the same lines of genes, although you can't see what they are, in these two bits of chromosomes, but they're two separate chromosomes. So some kind of recombination event has happened in the chimp chromosome to have that in two pieces, but you still have the same gene order in those two pieces of that chromosome. Okay, so in the regions of the genome that show a difference in DNA sequence between humans and chimpanzees, who actually has the ancestral DNA sequence? And these are the kind of questions that you can answer having the genome sequence of both of these. Okay, so essentially we're trying to answer, so which sequence was right, which sequence was the common ancestor sequence? Was it the human sequence at that point, or was it the chimpanzee sequence? Because there's a base change in that sequence. So let's have a look at an example. So we're looking at some gene in humans, and there's a T in a specific area of that gene. While in the chimpanzee, there's a G, okay? And so we're asking the question, which one of these, the ancestor of the human and chimpanzee, what did they have? Okay, so which is the ancestral sequence? Who changed? That's essentially the question we're asking. And how we can work that out, we can actually then we can compare the genomes of humans and chimpanzees with a more distantly related organism, for example, an orangutan. We call that an outgroup. So if you include an outgroup in this kind of analysis, you can see that the outgroup had a G at that position. So what we can say from that study is that the G was actually the ancestral sequence, because that distantly related organism had a G there, and therefore it's the human that changed that T form. And so you can do those, you can derive a sequence of a common ancestor doing these types of studies. So they're very powerful, looking at genome sequence of different organisms. Okay, so the last couple of slides, I just want to introduce this idea of a single nucleotide polymorphism, a single DNA base change, essentially what we were looking at there. So that would be the example of a single nucleotide polymorphism between human and chimp. So we've got a single base change, okay? So one DNA base change, and that's essentially the major difference between me and you. is lots and lots of these single DNA base changes. We call them SNPs for short, single nucleotide polymorphisms, and that is the difference when you compare two individuals. Okay, so there's lots of these SNPs. For example, individual 1 has a C at that position in the DNA sequence of some gene, and individual 2, instead of a C, has a T at that position. We would call these alleles of a SNP, of a single nucleotide polymorphism. So individual 1 has a C, so it's a C allele. Individual 2 has a T. And see, there would be 2 alleles of a SNP. You don't normally get more than 2 alleles of two changes in a single. You either have kind of one or the other. That's it seems to be a common occurrence with single nucleotide polymorphisms. But in any case, there's thousands of these when you compare if you were to compare my genome with yours, for example. So collectively, these very small changes are important, and they're important, and what we'll look at in the next slide, for understanding health, especially human health, and a susceptibility to disease. So you can also see now that we have genome sequences from, we're using human as an example, but genome sequences from humans from different populations, which was that more recent Nature paper that I talked about last week, where you have genome sequences, say, from 20 different populations and comparing them, okay? And then you can actually see the different single nucleotide polymorphisms that are a feature of that population compared to that population, for example. Okay, so when do we start talking about these single base changes as just random polymorphisms, and when do we start talking about them as mutations? Okay, so we carry this number, lots of these SNPs, and the majority of them are these single base changes, and we would share similar SNPs with family members or other members of a similar population. If this genetic variant, so this SNP is common, say common in a population, then it's not likely to be a disease mutation. Okay, so if it's very common in a population, maybe in that population, but not in that one, but in that population, lots of people have that single nucleotide polymorphism, isn't unlikely to have a severe effect on health because it's very common and most people have it, therefore likely not to be something that you evolutionarily keep. Okay, so it wouldn't be good to have a mutation. If a genetic variant, so a SNP, is rare in a population, but it's still there, it might be more likely to have a deleterious effect on health. So it's not common to have, but that's how we define a mutation. So if a mutation, if a single base change is rare in a population, we call it a mutation, and it's likely to have a health problem. So for example, like this TP63 that you're going to work on in the lab, this single mutation in that TP63 gene makes a protein that doesn't work properly, and you get this dysplasia, this developmental disorder. Okay, so that would be a very rare mutation that you would find in a population, less than 1%. And this is then how we distinguish between whether something is a mutation or a polymorphism. So it's just a normal change. It has a frequency that's greater than 1% in a population, doesn't normally cause a disease. And that's generally how we work these out. And a nice example, which is what I'm going to finish on, is a mutation. Actually, this would be a polymorphism because it doesn't confer disease. So it would actually should be called a SNP, a polymorphism. If mutation in the lactase gene allowed adults to begin to digest milk. And as this mutation or this polymorphism was beneficial, it spread through many populations. And you can actually track the mutation in this lactase gene to the establishment of agriculture and farming cows and people drinking milk. Okay, and so you can actually see this mutation spreading across populations because of the evolutionary advantage of being able to digest milk, where previously you couldn't. And so these are the kind of things that you can do with, again, sequencing data from lots of different organisms. So there's the learning outcome slide again. And then, this is the end of this lecture, and we'll continue talking about cloning and stuff next week in the next lecture.